why does tissue culture play such a pivotal role in modern science? That's the question we'll be delving into today. Tissue culture, or cell culture if you prefer, is a fascinating process. It's all about growing and maintaining cells or tissues outside their natural environment, under carefully controlled conditions. It's like a science experiment you might have conducted at school, but on a microscopic scale, with cells instead of chemicals. Now, there are several types of tissue culture techniques, and each has its unique features and applications. First up, we have adherent cell culture. This technique involves growing cells in a monolayer that's attached to the surface of a culture vessel, such as a petri dish or flask. It's a bit like growing a garden, but instead of soil, you're using a laboratory container. On the other hand, we have suspension cell culture. This is where cells are grown freely in suspension, not attaching to any surface. It's a common technique for cells that naturally grow in suspension, like blood cells or certain types of cancer cells. Imagine it as a zero-gravity environment for cells. Then there's primary cell culture, which involves cells that are directly isolated from an organism and cultured without being immortalized. Unlike the immortals of myth and legend, these primary cell cultures have a limited lifespan. That brings us to cell line culture, which is the opposite of primary cell culture in a way. Here, cells that have been immortalized through genetic manipulation or viral transformation are cultured indefinitely. These cell lines are the workhorses of research and biotechnology. We also have organ culture, where whole organs or pieces of organs are cultured ex vivo. This technique allows researchers to study organ development, function, and response to stimuli. It's like taking a peek at the inner workings of an organ, outside the body. Next, we have three-dimensional cell culture. In this method, cells grow in three-dimensional structures to mimic the architecture and function of tissues more closely. It's like constructing a miniature model of a tissue or organ, but with living cells. Last but certainly not least, there's plant tissue culture. Here, plant cells or tissues are cultured under sterile conditions. This technique is used to propagate plants, study plant growth and development, and produce genetically identical plants. It's a bit like cloning, but for plants. To wrap it up, every type of tissue culture has its own unique applications and advantages. Whether it's adherent cell culture for growing cells like a garden, suspension cell culture for a zero-gravity environment, primary and cell line cultures for studying lifespan and immortality, organ culture for a peek at the inner workings, three-dimensional cell culture for constructing miniature models, or plant tissue culture for plant propagation and study, each technique plays a vital role in advancing our understanding of the world on a cellular level. So the next time you hear about a breakthrough in cell biology, remember the vital role that tissue culture techniques play in modern science. After all, these microscopic gardens are where many of the secrets of life are discovered.